only Sunday talk scheduled for this year, and it's a new one for us because we have three speakers today. So this is uncharted territory for us, and I think we're all in for a very exciting talk this morning. Um, Most of you are familiar with how these talks go. If you have a question during the presentation, please type it in the chat. If your question is for a particular speaker, Alfred, Darren, or Sigrid, please specify who the question is for. If it's a general question, after the talk, we'll throw it out to our speakers and they can decide who's going to address it. We're going to mute everyone at the beginning of the talk, and please keep yourself muted until the end. And the talk is also being recorded and will be made available to the participating clubs in the next couple of days. With that, welcome to our speakers, Alfred, Darren, and Sigrid. Alfred Drummond-Herdman is based in Edinburgh, Scotland, and he studies the often overlooked but important group of coprophilous fungi. He uses macro time-lapse photography to reveal this world to a larger audience. He's currently working on taxonomic projects on the Bolbiteaceae and the Psathyrellaceae, including the description of new species and a revision of the genus. Darren Gordon is based in Dundee, Scotland, and has been studying fungi for the past 4 years. While he enjoys all types of fungi in general, his primary focus is on the Coprinoids, Inkcaps, and coprophilous species. His interest started when he began incubating herbivore dung, and he was amazed by the incredible fungal diversity that emerged. Darren particularly enjoys field work, looking for and identifying fungi in their natural habitats. He also helps manage social media platforms where he helps in identifying species from all over the world. Many of you are familiar with Sigrid Jacob, former president of the New York Mycological Society and a sequence validator for fundus, and for her work with the Ohio DNA lab. Sigrid became interested in dung fungi, I think 7 years ago, taking a class with Don Pfister of the Farlow Herbarium at Harvard on ascomycete fungi. It was a very dry summer that year, and Don suggested that people collect dung, and Sigrid got hooked. And over the past couple of years, she's described some new species of coprophilous fungi, notably a species growing on Canada goose dung, collected in New York City. She's particularly interested in species discovery, taxonomy, and making this kind of specialized knowledge accessible and available to a wider audience. And I'm really thrilled to have this great trio presenting to us this morning. So with that, Alfred, I'll hand it over to you.
Thank you. I'll just sort out the screen. Is this on the right page?
Yes, I can see the front page.
Brilliant. Well, thank you very much, Tom, for the introduction, and thank you, Tom and Juniper, and the rest of the consortium for organising this. I think I speak for Sigrid and Darren when I say we're always keen to talk to people about dung fungi, and try and share a bit of the what we love about it. So we are we're based slightly all around the world, but we meet up from time to time. This was at the All Things Fungi Festival, and we collaborate as well, in various projects. We are united by the interest in coprophilous fungi, and we're hoping that this talk is just going to provide the sort of the tops of the trees of this field, and just to show you why we find it so interesting, and hopefully why hopefully spread some of this. Now, John Webster, who was he studied coprophilous fungi in the sort of 60s and 70s, I think. This is how he started his address, and I think it's a good place to start a talk. Is that it's a bit of an odd topic, dung fungi, but stick with us, and we'll see where it goes. And another quote from Dennis. Now, this is talking about stale dog dung, and this is not actually what we're studying. This is probably the outer limit, but it's quite a fun quote just to contextualize the mindset of some of the people studying this. And this is just an overview of how we structured it. We expect this will probably take just over an hour. This is the shortest we could possibly make it. So with this, I'm going to hand over to Darren.
Hello, everyone. So with coprophilous fungi, a good question to start with would be, what are they? They're very simply put, they're dung fungi. They're fungi that inhibit or associate with the dung of animals. When studying dung fungi, it's important to look at the interaction between three kingdoms of life: animals, plants, and fungi. The animals eat the plants are digested, and the fungi grow on the resulting dung. Now, these fungi play an extremely crucial ecological role that essentially break the dung down and release vital nutrients back into the environment. Now, as you're about to see, fruiting bodies can vary considerably in size. Some of these are big, for example, Panaeolus semiovatus and Panaeolus subfirmis. Some are not so big. We'll have a suspected Chylomania rubra on the left and the lovely Narcissia scotica on the right, held up, grown from a piece of hair dung. And some are even smaller, like Sphaerobolus stellatus here with a pin for scale. It's a beautiful close-up. And even smaller than that is the fairly common Ascobolus immersus, also with a pin for scale. And we can get even smaller, like the Saccobolus. Now, here's an absolutely gorgeous example of Saccobolus beckii. Now, I wouldn't let Bjorn Wergen's beautiful photography skills fool you. This species is actually totally microscopic. And some of these fungi can even be single-celled, which Alfred will discuss a little more on further in the slides.
All right, let's get into the business of how the dung-loving lifestyle actually works. So if people take a look at this picture, most people say like that's just waste, but it's actually not waste at all, because herbivores only digest about half of what they eat. The rest is lignans and cellulose, and lignans and cellulose is exactly what fungi love eating, especially dung fungi. But if you are a fungus who likes to eat, who likes to feed on dung, if you're just sort of sitting there waiting for an animal to pass by to drop some, you could be waiting for a really long time. It's sort of like waiting for a bus. So what a few genera have figured out is like instead of waiting for dung to happen, you actually sort of colonize it before it even becomes dung. So you get ingested with the food, and then you hit the ground running. You're number one in the buffet line. So this is how the whole cycle works. So an animal, like a sheep here, will eat some vegetation. That vegetation will have spores from a dung fungus on it. It'll pass through the digestive tract. It will get excreted or pooped out. It'll sort of land in a pile of poop. Fungi will grow in it. The spores will disperse, and the animal will eat it. So that's how the cycle works. However, there's a number of challenges, and this is challenge number one. If you look at this picture of a meadow, you see all these darker green spots. What are these darker green spots? It's those are areas where animals have pooped, and animals won't eat where they've pooped, which is wise because there's parasites and all kinds of other nasty things in the poop. So there's sort of this thick barrier of grass around them. So the challenge for the fungus is like, how do you get out of that zone? And that zone is called zone of repugnance. So they have to figure out how to jump, and they have devised all kinds of strategies to do that jump. Ballistic ejection with adhesive spore packets. We're going to hear much more about that. They can hitchhike on insects. They could use deliquescence and ink as a method, or they could just elevate their fruiting structures and wait for the wind to blow the spores away, also known as a captain stem. So that's challenge number one. Challenge number two is like, it's a tough journey because like the gut is hot. It's full of digestive enzymes, and you have to survive all that. So a lot of species will either have really thick-walled spores, or they'll have a gelatinous sheath around the spore. And once you're out in the vegetation, the other risk is the UV radiation could sort of sterilize your spores. So a lot of spores use melanin sort of as a protective color, so that doesn't happen. So that's challenge number two. Challenge number three, lots and lots of other organisms also love to eat dung, from bacteria to springtails, nematodes, mites, fly larvae, and other arthropods. They all love a meal of dung. So fungi have to figure out how to come out ahead in that race. And it's not just all those other organisms, it's some of your fellow fungi too, because in a typical collection of dung fungus, you get well over 10 species, especially for marsupial dung or sheep and goat dung. Those are particularly productive. So there's a ton of competition going on. So what fungi have often do is they'll develop all these secondary metabolites like antibiotics, antifungals, antiparasitic agents, insecticides, larvicides, herbicides, just to sort of come out ahead in that warfare that happens on every single collection of dung. And actually, a lot of these have sort of secondary applications like pharmaceutical and agricultural uses. But yeah, it's a tough world in there. And then the final challenge is something that fungi really can't do anything about, but current farming practices do decrease the diversity and abundance of fungi and livestock dung. Obviously, that only applies to livestock. Many other animals are not impacted, but dungs these days get spread as slurry. There's grassland monocultures, there's more grain feeding, and a lot of animals are kept in barns and not fields, and all of these kind of decrease the abundance and diversity of dung fungi. So yeah, it's a tough world out there.
So I'm now going to talk a bit about succession. Succession is maybe a principle or a theory, or I'm not sure how you want to frame it, but it's to do with an order by which certain groups of fungi sort of generally observed to appear on dung, or rather when the fruiting bodies appear. It's not an exact science. There are, the textbooks might say this and that, and then you'll look at something and everything will be growing in the wrong way around. So there's a bit of nuance, but it's quite a helpful way just to think about it and to understand it. So first, typically the first few days or even the first day, you might get little things like this, this mucor, which is growing on some sheep dung. Maybe within the first week or so, depending on the temperatures, you'll get Pilobolus, which there'll be more on them later. And then you can get, for example, Chylomenia granulata, which can grow up in maybe the first 3 weeks, followed by some of the quicker growing Inkcaps. Then when you're entering more into the months, once the dung is it's completely sort of different to when it was just deposited, it's much more fibrous. You'll get these larger mushrooms growing from it. And really, they're not too, this could be earlier or later, but up to 6 months or more on dung, you can get these little ascomycetes. And so to illustrate this, for leading up to this talk, I've been filming some sheep dung and been keeping records of when each bit was filmed, so you can get an idea of the procession of the time. So I'm going to play this time lapse, and you'll see roughly this order at the beginning, or the first few weeks of what I've just talked about. So these Pilea grow very quickly, and they just elongate their stems. Now the Pilobolus is starting to show. And this was a surprise. I was trying to film something behind them, and this whole army appeared. And this is a bit more sped up, but you can start to see the ascomycetes growing. And finally, a bit more familiar to some would be one of these little Inkcaps that Darren's going to talk to us about. So we'll get started with the main groups then. We're doing this roughly in order of succession. So first, we've got the Mucoromycotina, or some textbooks will refer to them maybe as the Zygomycota. But the common name for them would be the sugar fungi, because a lot of these are eating the simple sugars that are very quick and easy to break down. Especially are found on fresh dung. Maybe it's partially stuff that hasn't been properly digested from the animal's digestion. And they're very quick growing. In a couple of hours or in a day. Some of them will be trying to make a very long stem to try and maybe lean onto a piece of grass or something nearby to get stuck. Some of them will be going up and it will dry out the little sac holding spores and maybe get blown into the wind. Some of them, and I think the most impressive, are the Pilobolus, which will, well, as I'm about to show you, they have this sac underneath the sporangium, which is designed to swell with liquid at great pressure. And it's also a lens. It will come up when the sun comes up in the morning, it will track it and it will follow exactly where the sun is to try and get itself up to a good angle to fire this little packet of spores away from the dung. And it's also it's actually I think it's the third fastest accelerating object in the natural kingdom, as far as I know. It's the speed of the tiny little black, and this is all, almost microscopic, it's a few millimeters tall, but the speed of it firing towards the sun, away from the dung, accelerates quicker than a bullet leaving a gun. Although the top speed is not that high, maybe 40 miles an hour, the acceleration is incredible. So this this slow motion video, which I'll show you here, not made by me, but that just shows you exactly how it works. I'll just play it. Try and play it again because it's quite quick. There's a huge amount of force has been left in there. So that was a slow motion video. I'm now going to show a time lapse, which is going to show it on the other way around. And this is showing their growth, and they are well. I'll let you have a look. So yeah, it's amazing, and it's all happening on lots of dung all the time, but just walk right past it, don't see it. This is something which I've just added in this morning, this picture. So some of these time lapses, I shot them in a sort of glass terrarium to keep all the conditions stable. And then I noticed, and it's just warmed up here in Scotland to about 10 degrees Celsius. It's almost tropical. But this has raised the temperature enough so that the spore packets that have hit the glass have actually started, by the looks of it, to germinate. And I've never seen this before on the terrarium, but it's pretty incredible. So here you can see that's the top of it, the wrinkled part, I think, and then the bottom part where there's that yellow gloopy material. That's what's in that balloon-shaped structure, and that will help it stick to a blade of grass. So it's just something quite a cool observation I've just seen. And this is not in the Mucoromycotina, but this is a really cool time lapse that we thought we'd just like to show. This is from Patrick Hickey, who does a lot of imaging, especially time lapse imaging of microscopic fungi or hyphae moving. And this is a remarkable fungus called Basidiomycota, which he cultured from frog droppings. And the time lapse shows something that may not have been observed before, which is it will create this single-celled, this is basically this nucleus that comes up with a single spore at the end of it, trying to wait for maybe a frog to pass over to grab it. And if it doesn't have any success, or if it doesn't like the conditions, Patrick's seen that it will actually send out an escape pod, this sort of escape hyphae, down from the base to the bottom, and then it will sort of cut the nucleus in half so it separates it. So you'll see that, and you'll also see it has another type of reproduction where it will actually shoot the single spore, which you'll see on the left. So just have a look at this. And with that, we're going to move on to the Inkcaps. So over to you, Darren.
So this is by far my favourite group of fungi. When studying dung, it's important to learn your Inkcaps or Coprinoids purely because they're an extremely common occurrence. Well, Inkcaps are a large group of fungi that originally belonged to the genus Coprinus. Further studies eventually split this genus up into several genera across two families. They're mostly recognised by their ability to deliquesce into the inky substance we all know and love, and also by the presence of veil or setiols. As I mentioned before, coprinoids is another term that you might have heard these referred to. It's a more scientific term and one I prefer to use not just to sound smarter, but also because not all Inkcaps actually deliquesce. And these species are extremely fast growers. Often they can emerge and complete their cycle within a day or two. And here's a simple look at how these splits leave the coprinoid genre looking now. On the left, Coprinus remains in Agaricaceae, and the rest were moved to Psathyrellaceae. We've got Coprinopsis, Coprinoids, Parasoa, and Narcissia. Now, there are other genus that are considered Inkcaps, but we'll be focusing on the groups that are well represented on dung. So Coprinus comes from the Greek word kopros, which literally translates to dung. So we're off to a good start. And these species emerge, they produce some of the largest species, coprinoid species on dung, and these emerge completely encased in a white veil that eventually cracks into these upturned fibrillose scales, eventually dispersing completely, leaving scattered fibrils across the darkening cap surfaces. So this is a species I found for the first time not so long ago, Coprinus sterquilinus, and I found it on its favourite substrate of horse dung. And unlike most Coprinus species, this one has an annulus that sits at the base of the stipe. But like most Coprinus and most Coprinoids, they're completely deliquescent, often leaving just a blob of ink at the apex of the stipe. Now, these caps will deliquesce from the margin inwards, and they do this as a strategic method to avoid dropping spores getting caught in the gills below. Another species to note would be Coprinus spediciosporus. Now, this is generally a smaller species and microscopically has much smaller spores, and it also associates with rabbit and deer dung as opposed to horse dung. Now, we'll move on to the largest genus of coprinoid species, which is Coprinopsis. And this genus contains some of the most commonly observed species on dung, and all of these species will present veil in a variety of shapes, textures, and consistencies. Now, species in this genus can be extremely difficult to identify. So often we'll break these down into sections where we can start to see some macroscopic differences. And we'll go through some of these sections and show some that are well represented on dung and some species that represent these sections well. So a good place to start would be section Narcoticae. An excellent example on the left here is the Coprinopsis stercaria. It's small, they're normally small, they're white to grey and completely covered head to toe in these velar elements. And if we look closely at the cap, we can see the cap is completely covered in these globose and sometimes subglobose cells, with an upper layer of the sparse filamentous veil. Now, these globose cells are strongly adherent to the cap surface, meaning they will remain throughout the maturity, whereas the filamentous elements often disperse and disappear completely. And this is a very, very common species. It's not fussy at all. It will grow on a range of herbivore dung, and this will often grow in large numbers. So a question I get asked a lot when it comes to incubating dung is, does it not smell? And to be honest with you, I don't think it smells at all. It could actually be some of the species that grow in the dung that could produce the worst smells. Here's two examples, Coprinopsis narcotica and Coprinopsis foetidella. Now, Alfred recently encountered narcotica on some dung and attempted a description which was a putrid, strong smell, even when dry, of old cigarette breath mixed with stale cat urine. Now, I hope that's not a smell any of us have encountered before, but it does give you an idea of just how potent these species could be, even in the presence of the dung they inhibit. Now, this is one of my favourite species. This is Coprinopsis sclerotiger. It's a rarely recorded species that was originally described from the UK. I've encountered this quite a few times through incubation, especially on rabbit dung and cow dung. So it's possibly quite common and maybe overlooked or just misidentified. Now, this one could be identified by the fact that it does grow from sclerotia, although it can grow directly from dung, which would make the identification process much more difficult. And another species that will grow from sclerotia is Coprinopsis tuberosa. Now, this tuberosa will often have smaller fruiting bodies, and you can differentiate these two also by looking at the spores under a microscope. So, what are sclerotia and what is their purpose? So, for me, I look at sclerotia as essentially a hibernation mode for fungi, and when they encounter unsuitable conditions like extremely cold temperatures or extremely long dry spells, the hyphae of the species will condense itself together and create this solid mass with a protective thick cell wall around the outer layer. And within these cell walls, they will store nutrients like glycogens and proteins that are vital for growth. And these structures will lay dormant for up to several months at a time and very patiently wait for better conditions to arise. And when these conditions do arise, they will produce fruiting bodies. And these structures can also produce new mycelial networks. So this just shows how well these fungi have adapted to survive their environments. So I'm going to show you a little time lapses, some species from Coprinopsis section narcoticae. It's potentially Stercaria, but these was never checked. And as you can see, there's plenty of sclerotia waiting patiently and lots of Stercaria growing gregariously. And they're always quite dramatic coprinoids, always collapsing. So for sections radiata and cinerae, I've decided to combine these two together purely because they share extremely similar characteristics, especially when we look at the veil, which is a very important feature. They often emerge completely white, covered in this shaggy fibrillose veil, and this veil is easily brushed or washed off. So for section radiata, we'll look at Coprinopsis radiata. Now these produce small fruiting bodies, although the stipes can become extremely long. And from my personal experience, I find this one most commonly on horse dung. And it doesn't just grow directly from the dung, this will also grow on the digested straw. And it's a strongly deliquescent species, meaning the caps and gills will completely deliquesce, and they do this at a quick rate. But here's a little time lapse of Coprinopsis radiata. Now, as you can see, the stipes grew extremely long, and like in typical coprinoid fashion, they like to do a little dance. And as the species matures, the caps will open up and turn up on themselves and begin to roll inwards. And this is a good method exposing the gills and spores to air currents, and eventually they, in typical coprinoid fashion, collapse into the substrate below. So a similar looking species, but a much larger species, is section cinerae, as Coprinopsis cinerea. Now, this species loves to grow in big dung heaps where there's lots of decaying organic material. But dung heaps are quite prone to drying out, especially at a surface level. So sometimes the species will grow deep into the substrate. Where it's so deep into the substrate where it's very dark and congested. So you'll often find the species growing from a rooting stipe or pseudorhiza. And this rooting stipe essentially acts as an anchor and pushes the fruiting bodies up through the surface into the light where it can carry on its life cycle. And this is another strongly deliquescent species. We'll show a time lapse just to show you that process. And as you can see, the margins begin to roll inwards and the caps will just, and gills will just completely deliquesce, leaving just a puddle of ink on the substrate. So for section niveae, the prime example would be Coprinopsis niveae. Niveae translate into white or snow white, and this reflects the colour of the fruiting bodies. And this is a very common species. It's probably one of the better known Inkcap species, especially when it comes to dung. The veil on this species is often described as powdery, and it often clumps together, creating these projections across the cap surface. And this one's more commonly associated with the dung of horses and cows. So a very closely related species to this one is Coprinopsis pseudoniveae. It shares many characteristics with niveae, but this one has these lovely pinkish tones to the cap surface. A much lesser known species is Coprinoids furfuraceae. Now, this species was described from China on sheep dung, I believe, in 2022. And this collection you see here was made by Alfred on a trip to the Isle of Colonsay. Me and Alfred actually took some time out to do some microscopy on this and couldn't reach a conclusion, so the samples were then sent to Sigrid for sequencing, and the results came back as Coprinopsis furfuraceae. As me and Alfred weren't actually aware of the species at the time, it just shows that we're still finding and discovering and describing new species all the time. Section Subniveae. So this is another one of my favourite species, Coprinopsis cercasiae. It's a beautiful species. If you look at the top right, you'll see what might look like a close-up of the alpaca's head, but it's in fact a young fruiting body, and it's covered in these beautiful brownish to almost golden fibrils. These fibrils will soon disperse and they'll reveal a white cap surface. So this species is close to my heart because it was first described from Croatia, I think it was in 2017. And the second recording was made by Alfred on some alpaca dung that I collected, some well-travelled alpaca dung that I collected and sent to him for observations. And not long after that, I actually found the species again on sheep dung. Now, it's possibly fairly common, and I would suspect it's easily overlooked. It looks very similar to other species of Coprinopsis, and it is, if you look to the next slide, it is also extremely small. So you've got to be very observant when it comes to these Inkcap species. So Coprinoids. Now for Coprinoids, we've got to look at section Cetulosae. Now this is quite a complicated one because species in this section were moved to their own genus, Tolozezus, which was later found to be illegitimate, and a new name was suggested of Ephemerosae, but for the sake of the talk, we're going to consider these still a part of Coprinoids. So species in this section are characterized by the presence of sechels or fine hairs on the cap and stipe surfaces. Now these hairs are often best observed through a hand lens and when the species are still young. Around 50% of species in this section are coprophilous, and although they mostly have sechels and fine hairs, some species can have some subtle velar elements, and these can vary greatly in size. So these are two observations I made. On the left is Coprinoids bisporus. This species matured to have a cap that expanded up to 30 millimeters across, and we'll have the very tiny, very small Coprinoids pellucidus on the right that was on cow dung and had a cap under 10 millimeters across. So this is a lovely little section in Coprinoids called section Hephemeræ. A great example of some young specimens on the left and maturer ones showing both the cap and gills on the right. So one of the species in this section is Coprinoids heptameris. And this is yet again another small species. This is quite unique in that it has both granular veil and sechels. As you can see here, especially when it's young, like most coprinoids, the granular veil or veil can often disperse. And although these are very small and very delicate looking, they're actually quite aggressive growers. These could produce secondary metabolites, called heptamerones, and these heptamerones can engage in physical combat through hyphal interference and can also produce chemicals that inhibit the germination of competing fungal spores, claiming the territory for themselves. As you can see at the bottom right, this is a collection of deer dung I made that I actually forgot about. Then when I looked again, I could see just this beautiful display of Coprinoids heptameris. And there's also a close lookalike to be wary of. This shows you how small these species are, and this is Coprinoids Psathyrellaceae, and these could be distinguished by observing microscopic characteristics. Another tiny species, as you can see from the bottom right, is Pilobolus. Now this one can be identified in the field if observing it through its full life cycle. It emerges with a bald cap, which is quite unique to the coprinoid world, and this cap has this beautiful reddish orange tone to it, that it loses as it matures, and we'll show this process through a time lapse. And last but not least, this is, in my opinion, and probably Alfred's opinion, a genus that contains some of the most beautiful species of fungi, not just on dung, but in general. It's Narcissia. So Narcissia, a classic Narcissia often emerges covered in this white granulose, almost sugar-like veil. And this veil will have an upper layer of more pigmented veil, often orange to brown, spread across that surface. And as species in this section mature, and the cap's fully open, they become extremely placate, and this veil will remain concentrated to the top of the ridges created and more prominent, especially at the center of the caps. And some of these species can display an annulus, for example, Narcissia hemorrhoides and Narcissia delicata. And as you can see at this close-up, of a beautiful translucent Narcissia stipe and the most delicate little annulus. That's probably enough ink caps for one day, so I'm going to pass you on to Sigrid for a deep dive into the world of Ascomycota.
Wow, they are gorgeous. Even as an Asco person, I have to admit, these are pretty stunning. Thank you, Darren. So yeah, now for something very, very different, we're going to look at the Ascomycota on dung. Finally. So yeah, I love the Ascomycota above all else, and mostly because the spore morphology is so insane. There's some of the most beautiful spores in the fungal kingdom are in the Ascomycota on dung. To those of you who don't know, what is an ascomycete? It's a phylum of fungi that produce spores in asci. What are asci? They're pressurized little sacs that function like microscopic cannons. Inside each ascus, the spores develop under some pressure, and then a trigger releases the pressure, and the spores are fired. And we're going to see in a second what that actually looks like. So yeah, there's so many genera on ascomycete genera on dung. This is a somewhat complete list. It would take days to go through all of them. And actually, Björn Wergern does a 4-day seminar where he covers some of those. So what I'm going to show you is just a small appetizer platter of genera, just to give you a sense of what's out there. So yeah, the two main types, basically, they either come as cups and discs, where they're somewhat open, the basidiomycetes and the liosiomycetes, or they come as flasks or fuzzballs where they have closed perithecia or pseudothecia, like the sodariomycetes and the theriomycetes. So yeah, let's look at some that have a cup or disc shape. So one of the most stunning examples is Ascobolus immersus. Some of you might have already seen it. It's very photogenic. It's also very tiny. You can see the asci with the spores sticking out. It's also a really fast flyer. It's actually even faster than Pilobolus. It's, I think, the highest acceleration of any species in any kingdom, I think. And yeah, let's look at a time lapse. So yeah, what you're seeing there is these gel-filled fingers that are sticking out and being ejected. And obviously what the gel helps with is adhesion to, vegetation. You kind of want to stick to a plant so you can get eaten. Ascobolus come in many shapes and sizes. They're usually either white or yellow. Some of the fingers are very small, like in Ascobolus granulatus, or they're a little bit larger, like in Ascobolus albitus over there on horse dung. What's most stunning about Ascobolus is also their spores. They're naturally purple. So this is not stained. This is just how they show up. And they have this beautiful ornamentation. Sometimes it's more stripy, sometimes a little bit more dotted, but it's always really stunning. Here's another example. Sometimes it shows up a little cracked, and then here's more dots. So yeah, these are all different kinds of Ascobolus. Related to Ascobolus are Saccobolus, and Saccobolus are even smaller. They're often just 0.2 millimeters across. They're either a yellow, they're purple, or they're white. This is from Björn. He did a little spread of this different different Saccobolus. These are extreme close-ups that I took. One is a probably undescribed species on left, a moose dung, and one is a close-up of Ascobolus citrinus, a very common Saccobolus. And once again, they've got a trick up their sleeve with regard to spores. These spores never come alone. They always cluster together. So one ascus will have 8 spores in them, but it's a cluster. And the clusters, once again, come in many different shapes and sizes. You can see some of them are strongly ornamented. Some of them are smooth, also different colors. Just a couple of examples, Saccobolus purpureus and rabbit dung. So these very condensed dark clusters, and then Saccobolus globuliferales on deer dung. They are not even colored, they're also black and white, and they're also very, very ornamented. Caelimania is a very common ascomycete on dung. Unfortunately, I've never seen it myself. It seems to not want to come down to New York City, but they look almost like tiny little eyelash cups. They have these setae at the edge of the cup. And then just as an example, you've got Theothecus, which I really like because it's so beautiful to microscope. But they show up as these little white barrels. And then when you scope them and dye them with phenyl cotton blue, you can see that the spores have these really cool polar appendages on both sides of the spore, which once again helps with adhesion to vegetation. All right, another one I really love seeing is Trichobolus zucalii. It's a sort of, I want to say like a little custard puff. The yellow in the center is actually a single ascus, and that single ascus contains up to 7,200 spores, and it's got these little spikes, maybe to protect against predation by insects. Okay, let's look at some of the flasks. Where the acai are sort of much more contained within a container here. There's a whole group of species, Podospora, Triangularia, Rhyophila, Pseudoecria, and Schizothekium. They're closely related, and they show up as these flasks, and they often have these, they have hairs or setae sticking out from the ostiole where the spores come out, maybe as protection. Some of them have little wings on the side, like Schizothekium there on the right. That's very characteristic of that genus. The best part of that whole family around Podospora are the spores, because they all have these appendages, these gel appendages, and they all look different. There's a pedicel and there's caudae, and there's sort of these gelatinous sort of appendages that, once again, help with adhering to vegetation. And if you look at them under the microscope, you can see them. It sometimes helps to stain them, like the example in the middle, but they all have these really neat little sort of attachments, and they all look different. Yeah, and that particular family is also really sort of extreme. Podospora gigantea has spores that are over 100 micrometers long. That's a tenth of a millimeter. So that's pretty large in the fungal kingdom. And some of them have tons and tons of spores in a single ascus. Podospora araniosa here on the right has 512, but there's also Podospora that have up to 2,048 spores in a single ascus. So that's pretty crazy. I love Delicia and Trichodelicia because they have, once again, neat-looking spores. They're very cinched at the waist. They're wearing a little belt. This is Delicia chodocola from Arizona that's also wearing a little fur coat. You can see it. The sort of perithecium is completely covered in these green hairs. And something closely related to Delicia is Trichodelicia. This one's heavily armed, the Trichodelicia minuta. It's got spikes everywhere, once again, I think to protect against predation. And spores have these cute little collars. You can sort of see them at both ends of the spore, these little bits sticking out. Sporomiella is by far my favorite genus. I adore this genus. The spores are so cool. So what you're seeing here is like single spores, but they're sort of separated into cells. Oftentimes they're just separated into 4 cells, but they can be separated into up to 16 cells. The fruiting body themselves are really boring looking, but once you pop them open, they can be astonishing. This is Björn photographing a few Sporomiella there on the left. Some of them have up to 16 cells, like Podospora herculea down there. So they're just like really neat looking. This is one of my favorites, the Sporomiella colomera. It's almost like a Michelin man. It seems to get thicker and thicker towards the middle. That's a 12th spore. And then last but not least, Chaetomium also has perithecia, but you can't really see them. They're sort of almost covered by these setae, these long hairs sticking out. They're very hard to ID. They can be human pathogens, so you have to be a little bit careful of them, but they often will fruit in great abundance on a lot of dung. So yeah, that's a little detour into some of the more common Ascomycetes. Also, I want to highlight here Peronia. Peronia is actually in Xylariales. It shows up more like a disc, and you can actually see it with the human eye without a microscope. The black areas on top of the disc are the areas where the spores come out, so the ends of the osteoles. This one's on horse dung from Arizona. The one on the right is it's anamorph, very elegant looking. That's it for Ascomycetes. We're back over to basidiomycota.
Right, we're now going to just finish the sort of the species groups sections with the agarics or actually not necessarily, just we'll call it the basidiomycota. So these are basically the fungi that reproduce by producing spores on basidia. So they drop the spores, the ones that Darren hasn't covered already. And this group, I'd expect if you were walking through a pasture or something, probably the ones you're more likely to spot without really trying will be these ones, because they grow generally the largest. So here we go. Lots of genera, a bit like the ascos. There's not as many as the ascos, but we've got quite a lot of different things that can grow on dung. But you'll be pleased to hear we will not cover all of these today. We'll just do a quick tour through some of the main groups. Panaeolus, or the mottlegills. These are probably the biggest, especially the species here, Panaeolus semiovatus, where I've seen cats sometimes, sort of this size, very large. And the name's semiovatus because of they look like half an egg. That's sort of the Latin translation of their name. The gills for Panaeolus will explain why they're called the mottlegills. And if we have a little closer look, you can see why they appear to be mottled. This is a cross section that I made of I can't remember which species in the genus. So showing some other features on the gill. But the main thing is to look at on the face, where you can see that there are mature spores organized in pores. But then there are also gaps, and these gaps are because they're maturing at different rates. So you can see that because the spores are dark when they mature and then pale when they're not, or when they're not there. So this is what gives it that mottled coffee stains look. Here are some quite large ones, all found in Scotland. And of course, I have to make a time lapse to really get their beauty across. If the pictures haven't done it, then hopefully this will help. It's also just worth mentioning all those water droplets that you see, they're not, that's not morning dew or anything. This is guttation that will be coming from within the mushroom, and it will secrete it out and it can suck it back up. It's a really cool feature. Next, conocybe or conocybe, however you want to pronounce it, which essentially means cone caps. There's quite a large genus. There's quite a lot of them are dung growers. And the main way to recognize these in the field is their orangey brown coloration. They're often quite slender, but they can also be quite stout. In the same family is the Bolbiteaceae, which is one of my favorite genera. It's very beautiful. They're probably not as sturdy as the conocybe. They could almost be sort of appearing to deliquesce in the way that they wither when they sort of at the end of their life cycle. And this hasn't been studied in as much detail, or rather, it's quite a challenging genus. Some of the groups are quite hard to distinguish. So this is definitely one if people are looking for areas to make some contributions. This one has a lot of work that needs to be done. Here's an example on a piece of Highland cow dung with lots of other species. This is Bolbiteaceae corrugatus, named so because of the corrugated cap. This is from Felipe in Spain, Bolbiteaceae elegans, which is a really cool species, which I think seems to be most common in Spain and that sort of area. And the cap you can see is completely, it almost looks like it's melted onto the stem. And this is a rare species called Bolbiteaceae excoriatus. And I found this is the first record in the UK. And there's not a huge amount of records in the world anyway, but yeah, it's a small one, quite unusual because of the brownish cap, but one to keep an eye out for. Conica, different family now. A very common one that you'll find to see in the field, and this is a worldwide species, so across the world there's a good chance that you can find Conica coprophilous. I've recorded a time lapse of these, but not a nice sort of naturey one. This is just in a plastic tub to show you also, this is dung incubation. This is what, when you're studying these up close and indoors, this is the sort of thing that you can see. and that was the cap wasn't turning black from any reason other than the fact that spores from its friends above it were dropping down, so it was being smothered by spores, and that's what caused that effect. Psilocybe, have to mention these, of course. These are those that contain psilocybin, very popular worldwide. In fact, probably one of the most well-known of species in this genus, Psilocybe cubensis, which is cultivated across the world, is a coprophilous fungus. But we don't get that in the UK. In the UK, we just get two types. We get Psilocybe pimentea, as you can see here, which is quite beautiful. It's got quite developed veil around the edge, which will help distinguish it from Psilocybe lineformans, which has much less veil development. This picture was taken in the New Forest in England, and I think they probably have more psilocybin in them. As you can see, that one on the right, the stipe has turned bluish. Psilocybe pimentea, I think there's a recent study that found in the ones they looked at, it didn't contain any psilocybin, probably just very variable, but very marginal. And lineformans, you can tell them apart in the field because it has these threads that you can just pull up with a tweezer on the gill edge, which is quite an unusual feature. Protostropharia, by far the most beautiful genus. I'm joking, probably not, but they're very hardy. These dung roundheads, they grow sort of all throughout the year, and you'll see them on cow dung, horse dung, lots of types, especially just in fields in temperate regions, and sometimes so it's quite wet, and you can tell it's them because you try and pick them up, and they're so slimy that your fingers will literally slip off them. You literally cannot pick them up. And so they have this mucousy layer around them that presumably helps to keep the water high and helps them not be picked. That must be the other reason. Psathyrellaceae, dangerous genus. Stay well away. Don't try and understand this. It's very complicated, and it's caused a lot of headache for me. But anyway, it's also very beautiful. So this is Psathyrellaceae hirta, which is very recognizable, and this is something you can recognize on dung in the field without a microscope. They do look a bit like sheep, which also helps to spot them. So they're not all the sort of mushroom-shaped ones. Some of them are a little bit more unusual, as you're about to see. So that was Pilobolus, the artillery fungus, and they sort of pop these spore sachets, spore packets out. Quite remarkable. This is the end of the species looking group of the talk. Now we're just in the final few bits where we're going to first talk about why we love studying dung fungi. So over to you, Sigrid.
Yes, so we all have our different angle on it, and I'm particularly excited by discovering interesting new species. And sorry about these very unappetizing shots of Canada goose dung, but which is a dung I don't normally incubate very much because it's not that productive. But I did a couple of years ago, and I got this kind of ugly looking black crust on them. And I thought like, ugh, and I was about to toss them, but I pulled up that crust, and under it were all these different, these little black spheres. And when I popped those little spheres, it turned out to be a spore myella. So that was intriguing. So I popped it under the microscope, and it was sort of 8 to 12 celled, and it looked, intriguing. And I compared it to keys, and I didn't really find anything it keyed out to, so I posted it on a German forum. And in that forum, somebody remembered that 20 years ago, Mike Richardson, a botanist mycologist, described something similar from Iceland, but didn't describe it as a new species, but he just sort of had a note about it. So I reached out to Mike. And he is a wonderful human being, and you can see him there on the right. He was been studying dung fungi for the last 50 years, so he knows a lot about a lot. And he said this is a new species, and we should describe it together. So I had to get the species that he found from an Icelandic herbarium and had to sequence it, and I sequenced my own collection and a couple of others. And then we described it as new. Joshua Bergebach over there on the right helped us with the phylogeny. Thank goodness, because I was still struggling with that at the time. And so, yeah, welcome to the world, Spormiella tela. Tela means loom, because it references this sort of strange carpet that this species is covered with. And it's been subsequently found a few more times, mostly in Germany. So it's a real thing. So yeah, Darren, what do you like about all this?
Everything. So hopefully what I love about dung fungi is everything this talk has represented. But for me personally, I love the field work and I love the connections that are made through studying dung fungi. I'm a naturalist. I love being out in nature. I love animals and I love engaging with these animals, especially when it's safe to do so. And I love the countless hours of just trekking through fields, stopping every 5 seconds to get up close and personal with every piece of dung I encounter. This is quite vital when you're looking for some of the smaller species, especially Inkcaps. And yeah, and I love the excitement I get when I see something I don't immediately recognize. And when I do, I'll often collect this dung, put it in a container, and what I love specifically about dung fungi is I could take it home and I could observe it from the comfort of my own settee. So in these winter months as well, when it's extremely cold and fungi are few and far between, I don't have to go far to go on a little foray through my tubs of dung. I also love documenting them, which is quite important if you're finding interesting or rare species. I love just taking measurements, etc., observing growth patterns and photographing the species through its full life cycle, which incubating dung allows you to do. I can't tell you how many times I had alarms set for like 3 a.m. in the morning, and I would have to wake up out of bed, go through, take a couple of pictures, and go back to bed ready and wake up soon after for work. And also by doing this, I could store and dry some specimens, bag them up, and send them forward to people like Alfred and Sigrid who could perform further research. It just kind of shows that from every stage, from field work to describing new species, it creates this big connection, and it just shows that everybody's just as important, and all the work that's done is equally as important. And last but not least, I love introducing people to dung fungi. I love seeing people's faces light up when I show them a piece of dung with a little mushroom on it, and it just opens up this whole new world of fungi to it. I love the expression when they see me carrying it with no gloves on, like I'm some monster. So yeah, just the whole process of finding the mushrooms and the connections that it creates through doing so.
So for me, the thing that I like, other than the time lapses, which I enjoy making too, but I love this the research side. It is extremely fascinating, very rewarding, and it's just fun to learn. Um, so I don't come from a biological background at all. I think I got a C at school when I was maybe aged 16, and that was the best. But so having to sort of learn everything is a bit of a process. But um, so this, but also collaborating with people across the world, I think that's one of the best things that I find about it. This was the beginning of one of the journeys. So this was a little mushroom that I found growing on Highland cow dung. Very small. I had no idea what it was. I posted it in some Facebook groups and sort of mixed results, but the consensus was that it was a member of Bolbiteaceae genus. Eventually did send some to Sigrid, who sequenced it, and then having looked at the DNA and got in contact with Katerina Malasheva from St. Petersburg, who's an expert in this genus. She thought this is probably a new species. So I went about to collect, find it more times because, and for some reason I hadn't found it before then. I haven't found it much now, but when I was looking for it, it would just grow all the time wherever I was looking. So it knew that I was trying to help describe it, and that's why it showed it to me. And we've recently, or a few months ago, published a paper describing this as new, which was a great international collaboration. Oops. And within this paper, I'm just using this as a bit of a Trojan horse to mention this this discussion that we had that we just like to draw attention to. It's not so we were confronted by trying to figure out why some of the mushrooms were very small. The first ones were very small, and then some of them were much larger. And there was this range, and it didn't appear to follow. We couldn't figure out why it was changing so much. Yet they were all the exact same species, and you think that we're suggesting that one of the factors that might affect this is the competition that's been discussed by Darren and Sigrid before. So if there's a more competitive species or a group of species, they might have sort of taken over more of the dung, and that could restrict the amount of space that this, for example, Bolbiteaceae has, which means this pool of resources or its sort of realized niche is very small, and as a result, it can only grow small fruiting bodies. You know, there will be lots of factors, but it's just trying to add an extra potential cause, and it's just fun to think about the processes in the dung because it's very active. Oh, and of course, yeah, I should have mentioned that this paper was the first time I think in history that a new species, probably of anything, but certainly of mushroom, was described with a time lapse as part of the official description. So I'm going to show you an extract of that time lapse now, and then at the end, and the video also will show a Conosobi and then another Bolbiteaceae. So that's the Bolbiteaceae. I'm also going to quickly mention my other sort of main area, probably actually my main area of interest, which started with Mike Richardson telling me that there are interesting fungi to be found on red grouse droppings and mountain hare droppings. in the Scottish Highlands and in the upland areas. So in 2024, I started collecting grouse droppings, as any normal person would do. And you can see on the left, in that little packet, I've collected some. This is in sort of heather moorland areas, often on quite a mossy ground. And after keeping some in some tubs for months, actually, some of them would remain motionless for months, but then sometimes these things would start to grow. And when Darren and I were first looking at them, we referred to them as the orange snowmen, and which is very scientific. But then we eventually set to work to describe them as a new species, which named the moorland inkcap, or Narcissia scotica. So this is another member of the genus Narcissia. And I did this with Laszlo Nagy from Hungary. There's another species. Did a little article with Pietro Botto from Italy, and we were looking at this species. I also found on red grouse droppings from Scotland. This is probably not the species we've named it, because actually it's probably a new species, but that's TBC. And there are a lot more Narcissia to be found. We've been sequencing a lot. Sigrid has been sequencing a lot, and lots of people across iNaturalist and other forums have been very helpful in sending us material, and we're starting to build up a picture of the genus. And as you can see on the right, this is a snapshot of the genetic, or sort of the evolutionary position of a lot of the species we've looked at, or the collections in the genus. And we can see it's very diverse. There's a lot more species in here that we don't know about yet. So this is underway. And to try and convince you that Narcissia is the most beautiful genus of fungi, I think it's best for me to end it on a time lapse, which will show a few different species.
Well, hopefully we've sparked some people's curiosities, and they'll be asking themselves, well, how did I get into this hobby of incubating dung? And as you can see, this hobby could become quite excessive and obsessive. So how does somebody take it further? Well, first of all, you've got to know where to find dung. A great place to start would be to go anywhere there's roaming herbivorous animals or grazing. And luckily for me, I've got surrounded by lots of farmland, so I've got lots of different dung available to me. But some people might not have that luxury. But suitable dung can be found even in the most urban environments. I often collect my squirrel dung from my local park. I find deer dung at the local golf course and plenty of rabbit dung at my local cemetery. So it's always good to know your dung. I'm probably better at identifying dung than I am fungi at this stage. So what's good dung would be dung from herbivorous animals, and which is great to collect and to incubate. What we're looking to avoid is the bad dung, which would be dung from carnivorous animals or meat-based dung. For three reasons. For one, this dung is extremely smelly. Two, it is not broken down by fungi. This dung is broken down by bacteria, and it's this bacterial decomposition that releases these awful smells. And three, because you're much more likely to encounter parasites. So when it comes to incubating dung, you're looking to get suitably sized containers. If you line the bottom of these containers with some damp paper towel and put the dung inside, try not to overload these containers with the dung, allowing them to breathe, and just spray this dung periodically to keep the moisture within the dung. Two useful pieces of equipment that you could use are a dissector and compound microscopes. These are extremely useful for looking at some of the smaller species like Ascomycetes and for looking at microscopic aspects like spores and basidia and ascus, for example. These aren't always accessible to people, and they're not crucial to learning about dung fungi.
Right. Okay, we're 2 minutes away from being finished. Thank you, everybody, for sticking with it. I know this is a little longer than the average presentation, but just a little bit more on resources. First of all, we do encourage people to post on iNaturalist because actually it allows us to see what people are finding, and oftentimes we'll reach out to people and ask them for their collections. So it's a really wonderful way of sharing your interests with other people. It also gives us a better idea of the ecology, the phenology, and the distribution of some of these species, and also will help identify what people are finding and vice versa. There's a beautiful network of global identifiers who can help you figure out what you found. If you want specific guidance on how to go about incubating dung, Alfred published this really lovely a PDF, and there's actually a QR code on this page, and we can share it later as well, that will connect you to a bunch of dung fungi related resources. Alfred, Darren, and I run the Coprophilous fungi Facebook group, over 2,000 members, lots of experts amongst them. We all have our social media, and there's also a really great German speaking forum called Pilz Forum, Coprophilous Filze, with lots of experts as well. And then there's some books available as PDFs. One is a massive doorstop of a book, Fungi Firmiculi Italici, very comprehensive, a lovely guide to dung fungi from New Zealand, but it covers a lot more. And then there's Mike Richardson's and Roy Watling's guide to fungi and dung. And yes, Mike Richardson, we want to dedicate this talk to Mike. Mike passed away last October. He was a great biologist and an even greater copromycologist. He spent the last 50 years studying dung fungi all over the world and writing some wonderful papers, not just about specific genera, but about dung fungi as a whole. I was very lucky to meet him last September. Him and his lovely wife, Barbara, who's also a noted biologist, in their sun-filled apartment in Edinburgh, and chat dung and travel and many other things. He was my co-author on my paper, and I owe him a ton.
Yeah, I'll also just say a few words about Mike. he was known to everybody in the dung fungi world. But he was particularly he was very kind. He was very good teacher and very good at happy to explain things to people. The first time I met him was this picture on the right at the last dung fungi seminar or workshop that him and Roy Watling gave in Edinburgh. And this really helped to sort of propel my interest in these fungi. And it was here that he had said about the interesting things to be found on the grouse dung. And so I wouldn't have found these new species were it not for him. But also he spoke a lot. He spoke a lot through email with each other. So he was very kind, very generous with his time in that sense. And him and Roy's keys remain, I still think, one of the best keys to dung fungi, because although it's technically outdated because of a lot of name changes, it's still very good for introducing you to the main groups. Anyway, we thought we'd dedicate this talk to him, and I think with that is us finally done. Thank you everybody for sticking by us talking about such a bizarre subject. If you have any questions, I'm not sure how this is going to work. I might hand over to
Tom will moderate the questions that were posted in the chat. So we'll just sit back, take a sip of water, and wait for the questions to arrive.
But first, a moment of applause.
That was amazing. Thank you so much. I don't know if you've been seeing these comments throughout the presentation of blown minds and shocked faces and hearts, and stunning presentation. Thank you so much. We do have a bunch of questions, and we'll get started now with Slava Makarov. You had a question for Sigrid. Do you want to unmute yourself?
Just before you start your question, though, Slava, I just want to say that we're going to go through and call on the people who put their questions into chat whose questions weren't addressed in the, like if we think that your question was addressed during the talk, we won't call on you, but then after we get to the questions in the chat, we'll have an open period. So if anybody wants to just jump in, then they can do so. Anyway, go ahead, Slava.
This is Kimberly. Slava had to run out to prepare for the big snowstorm that's coming. But Sigrid did respond, and he was okay with that, for her to get back to him. So that's fine.
Right. I just couldn't think of a good answer for your question. Maybe Alfred or Darren have an answer, but you were asking, like, there's obviously a lot of chemical warfare going on, but is there any kind of collaboration or sort of mutually beneficial relationships in the dung fungi? And I couldn't think of anything.
Well, that's a good question.
It is a good question. I mean, I'm kind of probably hyperfixated on the antagonism part, so I maybe am missing any signs of collaboration. But it's definitely a very dangerous place to be in the dung. I mean, I don't know if you're talking specifically about fungi, but I can't remember where I've heard this from. But there are certain in terms of dispersing spores, there are certain ascomycetes that will just appear in the bottom of, they'll appear just embedded in the dung, and they won't throw spores out, but they'll just sort of push them out, and they're hoping that the tiny little mites will run past and they'll get stuck to the mites, and these mites then go onto larger dung flies, and they crawl up the legs of the dung flies. So these flies then will go away to actually help spread the spores. There are probably, I'm not sure about symbiosis in the truest sense, but I'm sure somebody else on this call will know. So yeah, pass.
Next, Fred in the Philippines. You had a question. Do you want to unmute yourself, Fred?
Hi, good evening. Sorry, I'm very sleepy. But no, the presentation was wonderful. It's just really late here. I was gonna ask about about smell with regards to coprinoids and coprophilous fungi, because normally when we see them being requested for identification online, uh, people don't ever mention the smell. But if it's so prominent, you'd expect, right? You'd expect that people would think of them as more prominent. But at the same time, they're on dung. So how certain are we that the smell is definitely originating from these specimens and not from potential mishandling, or they're being in close proximity to other volatile stinky substances?
Yeah, it's a good question. So, I would start by saying, well, when you spend a lot of time around dung, you start to become familiar with the certain smells. In the cases of the two species I mentioned, Coprinopsis narcotica and Phoetodella, I'd actually came across seen Phoetodella a few times, but one thing that never popped into my head was to try and smell the mushroom to see if it did present any smells. And when Alfred found Coprinopsis narcotica, he couldn't underestimate how strong smelling it was, and he could be sure it was coming from the fruiting body for two reasons. These mushrooms are actually named for their smells. Narcotica representing narcotic smell, and Phoetodella meaning phoetid. And when Alfred detached the specimen from the substrate to dry them out for further studies, he said when he opened the bag, the smell was still extremely strong and present.
Okay, great. We'll move on then. Sigrid, oh, well, Craig, maybe you'd like to ask your question.
About the tools or about the safety?
About the tool.
Yeah.
Oh, sorry. Go ahead.
Ask any question. You got the mic. I don't worry about what you wrote. Just ask me what you'd love to know about.
Yeah, in terms of I've seen like these really small ones. I can see them under the microscope. When I try to like grab samples to put them under to see the spores, I'm getting a lot of like the dung as well. I'm destroying the samples. Do you just have any guidance for how to collect these?
Yeah, that's a common challenge, and honestly, it's not completely avoidable. But like, I use a very fine needle, and a needle on a stick, basically that's easy to hold, and I just use that to sort of gently lift it from the substrate. On a good day, only the specimen will stick to the needle and not the substrate. But yeah, definitely there's a lot of trial and error with that. No secret tricks.
Yeah, thanks.
Dawn Wayman, you had a question, and I think this is any of the three of you could jump in to answer this.
Do you want me to
Could just unmute
yeah.
yourself and ask your question.
I am unmuted.
Yep.
And I was gonna say, I just was curious if when you collected them, you froze them or put them in the refrigerator or dehydrated them to like speed up or slow down the process so that you had more time to do observations.
No, it's not something I would suggest doing. I think what we're hoping to do is to observe them, well, basically replicate the environment they would naturally grow in. So it's just you're just trying to make sure there's enough moisture, keep them in almost room temperature. And when it comes to photographing them and stuff, it's just a case of being extremely available and trying to judge almost how long the species takes to grow, how long it takes till the caps open. But yeah, the only thing I've ever froze is actually, as I talked about earlier, is this sclerotia. What I tried to do was to try and replicate some horrible conditions. Then I put them in the freezer for a couple of weeks and took them out into better conditions, and it actually started to get fruiting bodies to appear. But the dung itself, I would never freeze unless, I don't know. No, I would never freeze or put in the fridge.
Okay, and then did you dehydrate it whenever you send it over to Sigrid for sequencing?
Yeah, oh, are you talking about are you speaking about the samples we collect from the dung?
Correct.
Yeah. So it's with coprinoids it could be extremely difficult. You're wanting to dry them as quick as possible, and once dried, you're sending them off. I put them in a little plastic bag, which maybe isn't the best method,
That's good.
because it's wet. But you could use a silicone gel pack, and then I would forward the specimens. But yeah, specifically with coprinoids, you've got to be extremely quick.
Okay, thank you.
You're welcome.
And a related question from Porna. Would you like to unmute yourself and ask your question? I could just ask it. And again, this is a general question. Could you please guide us on how to preserve the specimens? Like if you're working with a little coprinoid, Darren, how do you dry it to preserve it to send off to be sequenced?
Oh, I'm actually going to pass this. Alfred's wrote a little guide on this, so I'm going to pass this on to him, and maybe he
Okay.
can forward that guide to some of the people that are. It's a bit more in depth than my explanation.
Yeah, so it is a challenge. I think one of the biggest challenges to studying the small, especially small ink caps, is that they just with a big a gust of warm air will just almost wither them. So um, I've put something into the chat, which I've written just a very simple guide on how to collect these really delicate ones. But essentially, what you're trying to do is use some tweezers and just take take them at the base of the stem, put them on maybe a sheet of paper in a warm room, warm, well-aired room, and if you just leave them for about 24 hours, you don't need to do any fancy fans or anything like that. If it's a warm room, they'll just dry out. And then put them straight into a plastic container. I wouldn't suggest at all putting them into a plastic bag actually, because that can crush them, and it can make it harder to look at them in a microscope later. So put them into like a little maybe a takeaway, a little sort of plastic takeaway sauce thing with a piece of silica gel sachet, and that they should stay like that for quite a long time. But I put a guide into a link into the chat, so hopefully that could help further.
It could also be useful to get these fungi just as the cap starts to open, because as soon as the cap and the spores start to mature, the deliquescent process will already start to begin, and that's when it becomes much more difficult. So
Mark Vicari, you had a question.
Oh, hi, yes. Thank you so much. Wonderful talk. It was really fascinating. I was wondering, okay, I live in a big city. There's lots of farms and horses that police ride and things like that. But I'm wondering if I get dung from, say, an animal that's been raised on grain, or if it's been eating hay or silage or something like that, is it still going to be as interesting to look at as dung from, say, a wild animal?
I mean, I can certainly speak to that because in New York City, I do see horse dung, but the horse dung is always from horses that, are being fed sterilized hay or some other kind of feed. And I have to say, compared to wild horse dung, it's a lot poorer. There are still a few species that regularly show up, including Ascomycetes actually, and Sordaria. So it's not completely lifeless, but I definitely find it to be a little bit less productive. But the fungi will find a way, wherever there's food, there will be at least one species or one genus who's figured out how to extract nutrition, even if it's less than ideal. That's my response, but maybe Darren and Alfred have different responses.
It's fascinating. Thank you so much.
Mhm.
yeah, I might add that as well. Obviously, especially wild roaming animals, they're exposed to like a lot more vegetation and stuff like that, and obviously more spores. So yeah, you're more likely to probably encounter a vast array of species on more wild from dung that's been secreted from more wild animals.
Right.
But I wouldn't let that put you off
Yeah,
collecting.
just go out, collect some, and tell us how you get on. See, there might be some interesting things.
Okay, thanks so much.
Marin, you had a question.
Hi, yeah. First, thanks all for the presentation. It was really fun. I just wanted to ask, like, is there any like record of anything growing on like insect rests? Just because it would be sort of generally abundant, though it wouldn't be like a massive sort of resource like a cow patty, but like I imagine something's got to be using it.
There are definitely things that grow on insect frass or dung. I seem to remember, I don't know if anyone else, Darren and Sigrid can, but in our Facebook group, somebody had posted a picture of stuff. I can't remember what it was. Certainly some of the mold-like things, the Mucoromycotina and things like that will be growing from them because they're very small fruiting bodies. But yeah, the answer is yes, stuff definitely will grow on them. I'm not sure exactly what.
I think not too long ago, I actually went to someone's house and they gave me some stick insect dung. So it's about as far as I went, and I found and was able to identify a species of Penicillium. So I can attest to that. But yes, nothing that overly excites me anyway.
Sure. Thank you.
So now, if anyone has any further questions, feel free to unmute yourself and just jump in.
I mean, there was definitely a few more questions that popped up recently. I know Osama had one, and Baird and Roman. I just want to make sure that those do get answered. It's hard to see because there's so much other commentary, most of it very positive. Yeah, I just want to make sure to, yeah, exactly. I want to respond to Osama. So yeah, do you want to ask your question?
Yes, first of all, thank you all for this interesting and wonderful meeting. It was a lot of many new informations for me and about the dung fungi world. So thanks you a lot. So my question for today, as a beginner, and you, Miss Sigurd, know about my story a little bit. So what I want, and this meeting has many mycologists. So I want your advices as a new beginner in the mycology world. What should I do, especially about dung fungi or any fungi? So I want your advices, please.
Yes, Osama, and you're already doing a lot of the things that will get you to becoming an expert. The first step is always to take really good field photographs, which you do. So yeah, really documenting a species, the species that you find thoroughly is a great start. And then the world of fungi is so overwhelming. You know, there's literally hundreds of thousands of described species. So it sometimes pays to specialize instead of trying to do all things. So maybe there is a genus that you are particularly attracted to, or that's particularly common in your area, and become a little bit of an expert, try and read some of the more recent literature about it, and begin to learn it and begin to get a sense of the small differences between different species of that genus. And with dung fungi, you know, like I've adopted my favorite genus, it's Spormiella. And you can ask me anything about that one genus. I'm not so good on some of the other ones. So going a little deeper rather than broad sometimes helps. But yeah, like posting things on iNaturalist is good because then you can also get feedback from other people. But yeah, I think you already your start is very strong.
Thanks a lot. And I will make sure that I will do exactly what you said. And actually, for my favorite species till now, it's Inocybraphiloids. You know, the first species we document and shared sequence. So yes, it's my favorite till now. You know, the first thing you discover have a place in your heart always in everything.
Thank you. Inocybe, my other great love. Yes, you have some amazing Inocybaceae in Palestine. Thank you.
You're welcome.
Reese, you had a question too. Do you want to unmute yourself?
Yeah. Hey y'all. One of my questions was I've came across Vavelia in the Gulf South region, particularly in Louisiana and Texas, Southeast Texas. Um one of my collaborators suspected that Vavelia might be an endophyte. How would we go about this question? Because I don't suspect we can sequence the grass that's inside the dung. So any insight on that?
It's
I'd appreciate it.
interesting. Yeah, and I've never tried this, but it would be interesting to actually sequence the grass and see if you do get something. I suspect if you use nanopore sequencing, which is better at pulling apart different species, you might actually get something, not with traditional Sanger sequencing. So nanopore might be worth a try. You just never know. But otherwise, the only other thing I can think of is like more like an eDNA methodology or something. But honestly, this is a question well above my pay grade, but it might not be a bad idea to try nanopore just to see what happens.
Might be worth, if you haven't already, contacting it's a German copromycologist called Peter Welt. I can put you in contact, but he sort of specializes in this group, so he might be able to provide some more specific advice or ideas at least. If you send me a message, I can pass it.
He's on Facebook as well, Peter Weld. He can be a little harsh, but he's very
Okay.
knowledgeable.
Thank you.
Okay, now, Bert, you had a question. Do you want to unmute yourself? Well, I'll read the question. Fungi on rats and mice are safe for study?
Who wants to take this one? I just want to remind people that the bubonic plague was transmitted by rats.
Further question about bat guano, which I guess could be
same response.
Yes.
Yeah, it's like bat guano is famous for carrying diseases that can impact human respiration. Yeah, there can be some very dangerous diseases lurking in omnivore and carnivore dung. Mice is a different story. Mouse dung is actually very productive and often has host-specific unique species. So I would definitely go with mouse dung, but I would run from rat dung personally. That's just me, though. Maybe Darren and Alfred have a different opinion.
No, I agree that. Sorry, go ahead.
Oh, I was going to say mouse dung can carry hantavirus.
Oh, shoot. Yeah, good point. Okay.
When it dries out and becomes airborne, it can
I'll take that back.
Cause pretty serious lung infections.
No mice, no rats, no bats.
Gene Hackman's wife died of virus.
I'd like to remind people of the good dung and bad dung section. The herbivores.
It might be worth quickly mentioning that there's a very interesting species that grows on mouse dung. If the virus is caused by it drying out, then maybe you have to keep it damp, which is good dung incubation practice anyway. But maybe best to stay away from it. But anyway, for wild mice dung. So if you look in a meadow or something, you can sometimes see these runnels, these sort of little paths that the mice make through the grass. And there's a fungus that has sort of adapted to work in this cycle, and I cannot remember the name. It's something like helicodendron or something like that. But it's microscopic and it creates these really cool, I think it's really cool bodies that are these strands and these corkscrews that come out from the side like this. And they're designed to get to snare the mice as they're running past. The whiskers are quite low to the ground, and they're trying to get caught on the mice whiskers. So then the mice will carry them somewhere else, and they'll preen themselves, and eventually the spores will end up in their near their mouth, and they'll end up in the dung. So there's a really it's just one of those cases of a fungus that's just adapted to a very specific niche, which you'll find in a lot of coprophilous fungi. This was something that Mike also discussed in his talk. I can try and find the name of the fungus. But yeah, in general, rats, I think, definitely stay away. It's not worth it. There's plenty of things to be found on cow dung are much nicer places. Mice maybe a little bit borderline, but certainly not house mice. I wouldn't do those.
Yeah, let's not get hantavirus, people.
Okay, now I think I think we've covered all the questions so far in the chat. So if anyone does have any questions or comments, would like to say something, feel free to unmute yourself and jump in.
Well, there's a question from Ben when we're coming to northern Sweden for field studies. As soon as you invite us, Ben, our emails are on this page.
Consider yourself invited.
All right, we'll talk about it.
That flight's paid for.
Have to do a GoFundMe.
I'll find some funding first, and then I'll send you an email.
Thanks, Ben.
Could I ask something very quickly? That first of all, the time-lapse videos are fantastic, just stunning. And I'm just sort of curious, it's not a major detail, but in a lot of the coprinoid fungi, we see a kind of wiggle as they are responding to something as they're growing, and they wiggle, they change the orientation of their caps. And I know that they are geotropic, but can you talk about what they might be responding to that's throwing them off like that? Or is that just something that just happens?
Oh,
sorry, I figured we no longer need that slide. We can just look each other in the eye.
So I'm not sharing my screen. Okay.
No.
Just a second. Okay. I'm discussing this recently. So the premise might be slightly wrong of the question, because you're saying that they're specifically like, why are they doing this? But maybe they're doing this. I know Patrick Hickey suggested that maybe they're trying to sort of weave through the grasses to get up. But it's also been suggested that would be placing too much emphasis on sort of adaptation. It could actually just be a result of that's how that's how they grow. So maybe they don't expand the stipes equally, but they're sort of these pulses of the compound that's growing them. I'm sort of going out of my depth here, but rather that they sort of develop it at different patches like this. So you're seeing what you think there must be a purpose for it, but actually that just could be simply that's just how the cells grow, and that's just how they go up. It's very interesting. It's not just the Inkcaps that do it. It's one of those things that the time lapses really reveal. They could also just be dancing, which is much more probably more likely. And they're sort of trying to be ballerinas, but they also have to spread their spores. Darren might have any other comments, maybe.
Well, yeah, I think you're maybe going down a bit of a scientific route there, which I'm not really qualified to talk on. But I wonder if it's just a case of they tend to have these really thin, fragile stipes, and as these stipes grow and become longer and longer, I think maybe the weight of the cap and maybe air currents or something might affect that, and it's causing them to almost go off balance. It's not uncommon to see actually coprinoids grow, collapse, and often pick themselves up again and continue to grow. I don't know if they do this through maybe sucking up water to build some solidity in the stipe again, but yeah, they're definitely a group of show-offs, and yeah.
Yeah, there's the turgor pressure, which is what's keeping them. It's a bit like when you have plants, if they're wilting because you haven't watered them, put them in water and they spring up. I think it's a similar thing whereby it's almost like hydraulics in these little Inkcaps. And I noticed once in one of my earliest time lapses, which was rubbish, and I will never show it, was I'd used I had cling film over the edge, that sort of covering the box that I had this alpaca dung in, and it was growing these little Inkcaps. And then I lifted the cling film off, and that let the warmer, drier air in the room rush in, and the time lapse was ongoing. And they all fall down because the humidity in the air has dropped, and that water, that sort of essential water pressure that's keeping them upright, had just been cut off. So all I did was I closed it, put a few sprays of a mister in it, And then you see, actually, maybe I'll show the time lapse. It's quite cool. And then you see them just pick themselves up again, as Darren said, and continue. So it's pretty remarkable.
Yeah, I'll just add as well to when doing time lapses, it's something I've attempted many times. And at one point I even had the same camera as Alfred, but I can't stress enough how difficult it is to actually follow these fruiting bodies perfectly and to create these time lapses. It's yeah, it requires a lot of patience and a lot of skill, and I've never quite been able to achieve the quality Alfred does. So I just want to, I think it's just cool, awesome to highlight just how amazing the work that he puts into it is. And yeah,
Thank you.
Coprinoids, like you say, they're just constantly moving. It takes a lot of predicting.
Any other questions?
It looks
Well,
like David, you're muted.
No, he's not, but go ahead, David.
Go ahead, David.
Yeah, so you're not muted, David, but we can't hear you. There's something with your audio connection that's not working proper. Sorry. You could put it into chat and we could read it out loud. Sorry, David.
In the meantime, I have a question about dung collected on unimproved grasslands rather than heavily manicured or grasslands where there have been human intervention, cemeteries, parks, golf courses. I know that something like Peronia punctata is something that was more common in the 19th century when there was more unimproved pasture land. Do you see differences in uh, fungi that you collect on dung and unimproved grasslands rather than urban or manicured grasslands?
I think they two are muted, so it looks like I'm answering this question. But I think you could definitely say for sure, safely say that it would affect the diversity of dung you're going to find. But I can also say from collecting, like I say, I've collected dung from the cemetery, and it's often rabbit dung I'm collecting. It seems to be one of the most productive, some of the most productive dung I get. It always starts with the Mucoromycotina, and I get these beautiful displays. Coprinopsis stercaria. It's where I find Coprinopsis sclerotiger, and I've never found. I found some species in Narcissia on it. I've never found that in incubation, but I do find that out in actual in the cemetery. So yeah, I think it's. I don't know the exact science behind it, but I would say it would affect it, but not to the point that you should not still incubate and collect dung from those sort of environments. Makes sense.
Yeah, there's sorry,
Oh, go on.
Oh, yeah, I mean, you can find them anywhere, and even if it's on sort of a very processed grain or sort of straw diet, somehow some will grow. But if you really want to maximize your chances, you really need to go to a place where there's the greatest diversity of plant of forage that you can find that the animals are eating. So so the unimproved grasslands, especially so the ones that we have in Scotland, that a lot of the things I study are highland cow dung. And these cows they're not even put on just for a few months, but they're sort of conservation grazed. So they're on the hill all the time, all year round. There's very little human input, just a little bit of locally sourced hay in the winter. But that means that first of all, you've got this very active cycle because it's a sort of unbroken cycle of eating, dunging, sporulating, eating, dunging, sporulating. But they've got this mixture of different grasses and heathery, shrubby, ericaceous plants, which will add a lot of fiber to the dung, but then also some of the things more like ryegrass and stuff. Having this diverse amount of plants means that there's sort of a greater amount of potential food for the fungi to have a chance to break down. And some species, you'll generally only find them in the very fibrous dung. So like the moorland inkcap, the Narcissia scotica, which I've described from heather moorland areas. This has been found only on the red grouse, mountain hare, and red deer dung. But the linking factor is that it's all animals that are eating heather, basically. And this creates super fibrous dung that certain things are adapted to and certain things aren't. So can't actually remember your question, but yeah, kind of waffled on about that. You can find them anywhere, basically, but especially on unimproved grass.
Did another question come in here, or?
Yeah, Fred.
Fred, did you want to unmute yourself, Fred, and ask your question? Maybe I'll read Fred's question. Oh, was that Fred?
Hi, good evening again. Sorry, I got by the internet a few moments ago. It just occurred to me because you're talking about the, English is not my first language. Okay, the dung preference for, certain fungi, they prefer a certain quality of dung, for example, or a quality of substrate. For example, in my case over here in the Philippines, we have compost piles, stuff like that. I tend to see section lanatule or cinerae fairly often in my compost piles. I know these also enjoy dung. I'm just thinking if, the fact that the availability of the substrate between, highly decayed stuff versus dung is enough to force a speciation between them. Or between these from the same section. Like the fact that in certain areas you're gonna have more dung, so the section lanatule there is, or cinerae there is going to be significantly different. Is it gonna be significantly different than stuff in the same region that are growing more on compost versus dung? I don't know if I'm making sense.
Yeah, I think you make perfect sense. I just don't think I'm qualified to answer that, to be fair. I think it would make a difference depending substrate to substrate, and depending on the nutrients that are available to the species. But to give a clear scientific response, I would not be able to do, unfortunately.
Would you mind just clarifying what the question was? You're talking about there's the difference between lanatule and cinerae and so on.
No, I mean, like, for example, if you have specimens, let's say in a farm of cinerae, for example, section cinerae specimens, and then on one section of the farm you've got lots of livestock, so they're growing out of the cow pile, and then another section it's the compost piles. They can also grow out of that. So I mean they're within proximity of one another, but because they are growing on different substrates over an extended period of time, is that sufficient to force speciation between those two populations?
So do you mean by speciation as in
like separate? Like if we were to because there's so many subsections and sections already for the coprinoids, for example. If we were to, for example, try to determine what species they are, like an very exact measure of identification between these two populations in the same area, based on the fact that they were growing out of different substrates. The population A is growing out of poop, the population B out of compost. Would that be sufficient? Over time, I guess I'm trying to ask if it's possible for us to use that as a model for evolution.
Right. Well, that's probably a question for Laszlo, Nagy, or somebody like that. I mean, as a comment, I think that these species could they can grow, they can sort of change a bit between. So there's the concept of strictly coprophilous fungi and or like or also obligate and facultatively, or there's different words, but some of them will just grow in that eating dying cycle, and they have to be eaten by the animal to go through. And some of them, like these ones you're talking about in the compost, will grow on the compost heaps. I don't know if that would be it. Probably, I can see it certainly being the case that. Well, the thing is, the compost heaps are slightly. I suppose they're slightly. They're just basically dung. There's not much difference between a compost heap and dung in a field, other than the amount of it that there is. So, in a sense, it's almost like compost heaps, almost like a species in there. If it's not already in the dung, then it must have come from elsewhere. Don't know if that would be enough over millions of years to cause speciation. I suppose anything would evolve over millions of years. Yeah, I'm probably also not qualified to answer this.
No, but let's just keep watching and see if they start to diversify. It's a longitudinal study, but let's get started.
You can maybe email it, and we can have another look.
I'd appreciate that. It's what you call it? It's something that's been on my mind because with the stuff that we have here, there are so many different substrates that are just popping up in different places in relatively close proximity to one another. So it can't help but think that we keep calling them bisections, right? So they're like different, but are they the same? Same but different, something like that.
Well, the sections are made on morphological grounds, predominantly, but also now there's the phylogenetic. Now we're in a phylogenetic age where a lot of them are supported by that. So the sections primarily are not to do with are they growing on this or that. Within Coprinopsis, at least, it's more to do with what does the veil look like? What does this look like? And then with some of them, when you follow it down to the habitats that they inhabit, then they might have a preference here or there. But it's primarily the sections are morphology based.
Okay.
Just
thank you. By the way, thank you.
And for
Interesting
everyone's information, Alfred has put his and Darren's and Sigrid's email in the chat. If further questions come up, you should feel free to get in touch with them. And someone had asked earlier about the recording, and
they've reached out to me, and I will share the recording with them.
Yeah. So it was recorded. The recording will be distributed to the participating clubs. We'll also supply Alfred and Darren with recordings. And if you ended up here from a link that Alfred or Darren shared, they'll be able to supply the link to you. A really amazing presentation. Thank you all so much. And thank everyone for coming, spending your Sunday learning about dung fungi and seeing these just spectacular time lapse of these incredible fungi that I think most mushroom hunters walk by every time they're out in the field looking for mushrooms and don't notice. Thank you so much.
Thank you for hosting us as well. And thanks everybody. Hopefully we've shown you that there's a hidden world just at your fingertips or at the edge of your dung tubs. So get incubating and see what you can find. Thank you.
Thank you very much, everyone.
Thank you.
Thanks. Thanks a lot. Great to see everybody, and see you next time. And thanks so much to Sigrid, Alfred, and Darren.
Thank you all.
Thank you. It was great being able to present.